In this video, we are going to be talking about the concept of cleavage. In the previous video, we have already introduced ourselves to the concept of embryology, and we said it is the study of development of embryo from a single cell to a multicellular organisms. And we have to understand that the process of cleavage remains one of the earliest mechanical activity in the conversion of single cell egg into a multicellular embryo, and it is initiated by the sperm during fertilization. However, in parthenogenetic egg, cleavage can commence without the influence of fertilization actually. Now, you see the process of cleavage. It can also be called cellulation. So I can say cleavage or cellulation happens through a repeated mitotic division. So it happens through a repeated mitotic division, like rapid succession of cell division. So these result in cells. So the mitotic division results in cells called the blastome or the blastomeres. Now, each of these cells, they occupy different region and differentiate into different type of body cells. Now, the end result of cleavage, like the end of result of cleavage, I think I've mentioned that yesterday, is called the morula. That's the end result of cleavage. And this morula is developed into something we call, it's transformed into something we call the blastula. You understand? And the wall of the blastula is called the what? Is called the blastoderm. And it has a central cavity called the what? Called the blastocell. I've shown you how that one looks yesterday. In the last video, that I dropped, I've already explained those ones. Now, we'll first start with what we have said yesterday. It is a process of progressive subdivision of zygote by mitotic cell division into an increasing number of cells of progressively decreasing in size, and it results in what we refer to as what? Morula. Now, I've already mentioned that it is a biphasic cell cycle and it has only two phases. That is the mitotic phase and the synthetic phase. Why? Because the cells does not grow, like the whole volume of the cell remains constant. That is one of the peculiar characteristics of cleavage, like the size of the cells remains the same, it does not change. You understand? So we have already mentioned that yesterday, and we said part of the characteristic of cleavage include the unicellular fertilized egg is transformed into a multicellular cells. There is no growth during cleavage. The general shape of the embryo does not change, and there is no formation of cavity in the interior. There is a formation of cavity rather. There is a formation of cavity in the interior that we know as the blastocoel, and the constituents of the cytoplasm are not displaced. And I think I illustrated how a blastocoel looks like and how a blastula looks like after the end of the cell division called the cleavage. And I also mentioned the peculiarity of cell division in cleavage. Also, I mentioned that temperature affects the time interval at which the cell increases. For example, I use goldfish, frog, and mammals, where goldfish cleaves every 20 minutes, frog every 1 hour, and mammals every 10 to 12 hours. I've mentioned all those ones. So today we'll be looking at deep into the concept of cleavage. Now, you see, the mitotic promoting factor complex is what it is what initiates the concept of cleavage, and this mitotic promoting factor complex is very very high during the mitotic phase, but it is undetectable during the synthetic phase. You understand? Now, this mitotic complex factor contains of two proteins. It contains of what? Of two proteins, and that is cell division control protein, that is cyclin dependent kinase, that is CDK, cyclin dependent kinase, and cyclin itself. So these are two protein that is called the mitotic promoting factor. That is what is promoting the division, like the rapid division of cells. You understand? Now, this mitotic promoting factor enhance or promote entrance into mitosis. You understand? It promotes entrance into mitosis. Now, cleavage is actually the result of two coordinated process. And what are the two coordinated process? That is karyokinesis and cytokinesis. I mentioned this yesterday. What is karyokinesis? That is division of the nucleus. And what is cytokinesis? That is division of the cytoplasm without the separation of the cytoplasm, which means cytoplasm divides, but it does not separate because they keep locked in into each other. You understand? We have something like this. Afterwards, you're going to have something like this. It is not going to separate into 4 cells. It is just going to divide it, but it's not going to separate from each other. That is basically what the concept of cleavage is. So it's happens due to two coordinated process. That is the karyokinesis, which is the division of the nucleus. The first thing that happens is the nucleus divides, and the second thing that happens is the cytoplasm divide, called cytokinesis. Now let us talk about the planes of cleavage, because understanding the planes of cleavage is very, very important, especially when we want to talk about the types of cleavage itself, where we're talking about the holoblastic, the metaplastic, the radial, the spiral type of cleavage. You understand? So let us get the concept of the plane of cleavage first. Now, the first one is meridional plane. Now, the plane of cleavage lies on the animal vegetal axis and it bisects both poles of the egg. Thus, it divides the egg into two equal halves, which means it divides the eggs. The cleavage takes place from the animal pole to the vegetal pole. Now, the question is, what is animal pole? What is vegetal pole? You see, the part of it is usually at this upper part is the animal pole. You see, the part that has less yolk, like it has less yolk, and it is very, very active because of the presence of nucleus, is called the animal pole. You understand? You now see the part that is having more yolk, like it is having abundant amount of yolk, usually at the lower part, is called the vegetal pole. You understand? We call it the vegetal pole. So dissecting an egg from the animal pole to the vegetal pole is what we refer to as the meridional plane. You understand? And this happen in Amphioxus and frog. And when we get to the cleavage in Amphioxus, you will understand more of this, especially when we're talking about the equal holoblastic cleavage. Now, the second one is vertical plane. Now, vertical plane takes place from the animal pole to the vegetal plane. Now, it happens, the difference between meridional plane and vertical plane is that it happens away from the center, unlike the vegetal meridional plane that happens at the center. This one happens away from the center, but it also occurs at the vegetal pole from the animal pole to the vegetal pole, and it occurs in furrows of chicks and also in frogs. So it divides the cell into unequal halves. Now, the third one is the equatorial plane. Now, in this equatorial plane, the cleavage furrows passes from the equator of the eggs. You understand? This cleavage plane bisects the egg at right angle to the main axis. Do you understand? So it lies on the equatorial plane. Therefore, it divides the egg into two halves, upper halves and lower halves, not right and left, upper and lower. As you can see, it is around this place. And it also happens in the third cleavage of Amphioxus. You understand? This one happens in the first cleavage of Amphioxus. This one happens in the third cleavage of Amphioxus. You understand? Now, the next one is the latitudinal or the transverse or the horizontal plane. Now, this is similar to the equatorial plane, but it lies on the other side of the equator. You understand? Just like the vertical is similar to the meridional plane, the latitudinal is similar to the equatorial plane, but lies on the other side of the equator. It does not passes through the center. You understand? So it is also called transverse or the horizontal. So that these are the planes of cleavage. You understand? Now let us now talk about the patterns of cleavage. That is where we are going to go. You understand? Now, they said cleavage has a special relationship to the egg region established by the cytoplasmic movement, and the position of the first cleavage is not random, but tends to be specified by the point of sperm entry and the subsequent rotation of the egg cytoplasm. Now said the coordination of the cleavage plane and the cytoplasmic rearrangement is probably mediated through the microtubule of the sperm aster. That is what initiated the patterns of cleavage itself. But that is not where we are going to. Let us just know the types or the several cleavage patterns that we have. The first one is the complete cleavage. Now, complete cleavage, that is also called the holoblastic. The cleavage plane divides the egg completely. You understand? It divides the egg completely, and it is further divided into bilateral cleavage, radial cleavage, rotational cleavage, spiral cleavage, which we will be talking about in this particular video. We'll still continue with it in this particular video, trying to look for an image to back that up. Well, let us just proceed. But we have to understand that a complete cleavage is when the eggs are completely divided, and they are referred to as what? They are referred to as holoblastic cleavage. Now, this holoblastic cleavage can be found in echinoderms, marine annelids. It can be found in marine annelids as well. Now, let's talk about the bilateral cleavage. Now, in bilateral cleavage, the first cleavage results in the bisection of the zygote into the left and the right half. You understand? Into the left and the right half. The following cleavage planes are centered from the axis, and the two halves of the embryo are mirror image of each other, and that is what we refer to as the bilateral image, and that's what we refer to as the bilateral cleavage. I'm trying to look for the image that shows the bilateral cleavage around this place. Let me see. I think it is here. Let us see. Now look at this. Coming, you can see it. But this is holoblastic cleavage. Look at it. The first one happens what? Along like meridional plane. You understand? That is why we have to take the pattern of the planes of cleavage first. Look at it. If you look at it very well, this side is the mirror image of this side. You understand? Therefore, after it divides like this, it now divides like this to give what? To give 4 cells. And then it divides like this to give what? To give 8 cells. So if you look at it very well, the first thing that happens is the what? Is dividing through the meridional plane, right? Then dividing through the equatorial plane. That is how it happens. You understand? And it is found in amphioxus and the echinoderms. You understand? Now we have the second one to be the radial cleavage. The radial cleavage in this pattern, when the organism is viewed from the above, is essentially radial in symmetry, and this is a characteristic of deuterostomes, some vertebrates, and echinoderms. Like deuterostome are those animals that their anus is formed before their mouth. When we get to gastrulation, we understand more about how the anus is formed before the mouth itself. Now we can see the radial cleavage around this place. You see, this is the radial cleavage. Firstly, it divides like this, like around the meridional plane. Sorry, around the meridional plane, then secondly around the equatorial plane. You understand? So this is what we refer to as what we refer to as the radial cleavage itself. Now this third one is the spiral cleavage, referred to as spiral cleavage. Now I'm trying to look for an image on this spiral cleavage. Now you see, spiral cleavage occurs such that the resulting daughter cells are not located exactly on top of one another. Like, they are not located exactly on top of one another. Instead, they are located at a slight angle. So spiral cleavage is a characteristic of protosome and results in determinant cells. There's something we call the determinate cleavage and the indeterminate cleavage. We still get there. So they result in determinate cells, and determinant cells are cells that their fate, like their embryological fate, has been determined. You understand? It has been determined during their earlier development of the embryo. So in other words, determinant cells are programmed to become a specific cell early during the process, which means if you look at a determinant cells, you would have known what they will become. And we have an example, we have an image for a spiral cleavage here. You see that they are not exactly on top of each other. So it is found in annelid and mollusk. You see that they are not found exactly on top of each other. You understand? So it is going to be slightly at a particular angle to each other and that is what we refer to as what we refer to as spiral cleavage. And the cells that are formed during spiral cleavage are determinant cells or determinate cells. That their fate has been determined. You understand? So that is what a spiral cleavage is. Now, the next one we have here, I've explained spiral cleavage, and as you can see around this place, they are explaining the complete cleavage, right? They are explaining the complete or holoblastic cleavage. Can you see that all of them cleave like completely? You understand? So that is what this image is trying to explain. So we have complete cleavage depending on the distribution of yolk. It is further divided into the holoblastic or equal cleavage and the holoblastic unequal cleavage. Now, the holoblastic equal cleavage happens when the amount of yolk is less or moderate and distribution of yolk is even. Like when the amount of yolk is less, but the distribution of the yolk around the egg cytoplasm is even. Now, the cleavage furrows divide the egg completely into equal sized blastome, and this blastome lies above each other. So the resulting cleavage is holoblastic equal, like equal holoblastic cleavage, and it happens in amphibians. So we also have the holoblastic unequal cleavage, and this happen when the amount of yolk is large, but the yolk is not evenly distributed. And what can happen that the yolk will not be equally distributed is that there will be less yolk at the animal pole, like I've explained to you. Animal pole is going to have a less yolk, and vegetal pole is going to have what? Is going to have more pole, more yolk rather, more yolk. You understand? More yolk, and animal pole is going to have what? Is going to have less yolk. So the cleavage furrows divide the egg completely, but they are of unequal size, and that is what we refer to as holoblastic unequal cleavage. You understand? So that is what happens in amphibians, example frogs. Now we now have the meroblastic cleavage. Meroblastic cleavage, the egg having a large amount of yolk, partial cleavage is observed, which means meroblastic cleavage is also referred to as something like a partial cleavage, like there is no complete cleavage of the egg. You understand? And we have different types. We have the meroblastic discoidal cleavage, in which the cleavage furrow does not penetrate the yolk, and this embryo forms a disc of shell at the animal pole where the cytoplasm is present, and this disc is formed at the top of the yolk called the blastodisc. And I would love to show you. I think I have this. Look at this. This is what I'm trying to say. If you look at this very well, this is an incomplete cleavage. So if you look at it, this cleavage furrows does not pass through the cleavage, like through the cytoplasm of the egg itself. It is concentrated at the center. You understand? So they form this particular cell. They form this particular stuff, and we call it the what? Can you see? Something like this. So we call it what? We call it the blastodisc. So the embryo develop on top of the yolk as a disc of cells, and we call it what? We call it blastodisc. So that is what we refer to as the cleavage is incomplete. You understand? And it happens especially in chickens. You understand? So we now have the meroblastic superficial cleavage. You see, in meroblastic superficial cleavage, nuclei divide by mitosis, but there is no cytokinesis, which means there is no division of the cytoplasm at all. That is, many nuclei are present in a single cell forming syncytium. For example, instead of us having something like this as cytokinesis, we are going to be having cells, 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 everything like that. You understand? Like all of them are just like that without the division of the cells, and that is what they are trying to like show here. And it happens in insect like Drosophila melanogaster, that is fruit fly. And what is it? Multiple nuclear division and result in the zygotes of many nuclei without cytokinesis. Like there is no division of the cytoplasm, but there is division of the nucleus. Like nucleus divides without the cytoplasm being divided. Now, the nuclei, like the nuclei that is being formed, now migrate to the periphery of the plasma membrane. You understand? They migrate to this periphery of the plasma membrane. You understand? And it happens in insects like Drosophila melanogaster. That is an example of an insect that this one occurs. Now we have determinate cleavage. I think I've mentioned that we are going to talk about that before. Determinate cleavage, it happens when the cells, like the developmental phase of each embryonic cell, has been established very early. Do you understand? So if you have a cell like this, maybe we can say that these are the cells. You see these cells, the fate of this cell has been known initially. That is what we refer to as determinate cleavage. Do you understand? And if it is an indeterminate cleavage, it means the embryonic cell's fate is not known. Do you understand? Embryonic cell's fate is not known. That is what we refer to as indeterminate cleavage. And determinate cleavage occurs in example Ascaris lumbricoides and some mollusks. And indeterminate cleavage occurs in vertebrate embryos. You understand? So these are what we have in the types of cleavage itself. So it can be classified into determinate and indeterminate types. It can also be classified into real cleavage or spiral cleavage. It can also be classified into holoblastic cleavage or meroblastic cleavage. You understand? So I can say that cleavage can be classified into holoblastic, sorry, into holoblastic or meroblastic. It can also be classified into radial or spiral cleavage, and it can also be classified into determinate or indeterminate cleavage. You understand? Now, these holoblastic and meroblastic can now be classified into either equal holoblastic, total holoblastic, or like equal holoblastic or unequal holoblastic. The meroblastic can be classified into discoidal or superficial. I've explained all that. Now, there are now some laws that backs up the concept of cleavage. So these are what we are seeing here. Let me quickly show you these images first before we proceed. So this is an isolecital egg. I've explained what an isolecital egg, those ones that are having evenly distributed yolk. And we have the holoblastic complete cleavage, where we have the radial, the spiral, the bilateral, the rotational ones. And we also have the telulecital egg. I think I have the full image here. So we have the meroblastic cleavage, where we have the telulecital egg can undergo the meroblastic cleavage, and I've shown you before they form something we call the blastodisc. And we have the discoidal. This is another one. This is bilateral. The bilateral cleavage, this is discoidal, and this is superficial cleavage. And what happens in superficial cleavage is karyokinesis without cytokinesis. That's the division of the nucleus without the division of the cytoplasm. You understand? Now there are now some laws that backs up. That is what we are trying to show you. There are some laws that backs up the cleavage patterns. You understand? There are some laws that backs up the cleavage patterns, and I'll be stating only two laws. That is Sachs law, and the second one is Balfour's law. Balfour's law. Sorry, Balfour's law. So this Sachs law was proposed in the year 1877. In the year 1877. So what does it state? The Sachs law states that the cell tends to divide. The cell tends to divide into equal daughter cells. Into equal daughter cells. Let me just say DC. You understand? And each, that's the first one. And the second one is like B for Sachs law. It says that each new division plane tends to intersect the preceding plane at right angle. The preceding plane, sorry, at right angles. That is Sachs law. That is what Sachs law. Now the second one is Balfour's law. Balfour's law now states that what? Balfour's law states that the speed or the rate of cleavage, that is the cleavage per unit time, like the cleavage, the rate of cleavage in any region of egg is inversely proportional proportional to the amount of yolk. You understand? It is inversely proportional to the amount of yolk, which means the higher the speed, the lower the amount of yolk. That is what it is trying to say. Do you understand what I'm trying to say now? So if you are asked, remember I told you yesterday that we have four types. We have the mesolecital egg. Sorry, we have the hololecital egg, abi homolecital egg, that is iso, homolecital egg. We have the mesolecital egg. We have the telolecital egg, and we have the centrolecital egg. Now remember, this one is evenly distributed, this one has moderate, and this one has enormous. So if they ask you which one is going to take more time, like which one is going to take more time for it to divide, for cleavage to happen, that will be the one with the enormous amount of yolk. You understand? That will be the one with it, because the rate or the speed is inversely proportional to the what? To the amount of yolk. You understand? In cleavage. You understand? So that is what the Balfour's law states. So in the next class, we'll be talking about the chemical change during cleavage, and I'll be showing you an example of cleavage, especially in mammals, in humans, the one we can relate with. That is what I'll be showing you, and I'll be explaining the concept of blastulation in the next video, by God's grace. So see you in the next video. Thank you.